Morning, everyone. My name is Clara Lajean-Sharon. I'm the Deputy Director for the UCLA Institute for Precision Health, and I'm really excited that I get to introduce my dear friend and colleague, Dr. Paul Boutros, and his really exciting talk on the origins of cancer aggressiveness. A little bit about Paul, which he's cringing in the corner right now because he really doesn't want me to read this. But he's a professor in the departments of human genetics and urology here at the David Geffen School of Medicine. He's also the director of cancer data science for the Jonsson Comprehensive Cancer Center. He's associate director for cancer informatics for the Institute for Precision Health. And he most recently serves as the interim vice dean of research for the School of Medicine. By way of background, Paul earned his bachelor's from the University of Waterloo in chemistry and a PhD from the University of Toronto in medical biophysics. So not a silly boy, or maybe he is. While at Toronto, he also earned an executive MBA from the Rothman School of Management. And in 2008, Paul started his independent research career at the Ontario Institute for Cancer Research, first as a fellow and then as a principal investigator. And then he moved to California and graced us with his presence and joined the faculty here in 2018. He'll talk about his research. It really lies at the intersection of clinical, molecular, and imaging data. He focuses on how these diverse pieces of information can be linked to personalized therapy for cancer patients. And by applying techniques from modern data science strategies, particularly machine learning, he creates biomarkers to help ensure therapies are selected that maximize cure and minimize morbidities. So without further ado, Paul.
Thanks, Clara. Now I get to be embarrassed. All right. So thanks, everybody, for coming, either in person or online. Oh, yeah, we got lots of people online. So I'm going to talk to you about effectively work that I've been doing my whole career, trying to understand why some cancers kill and lots of cancers don't. So over there, cool. All right, a couple of disclosures, scientific advisory boards, none related to what I'm going to talk to you about today. So this is the story we're going to go through. The clinical space in which I work, then kind of think through the central dogma: DNA, epigenetics, RNA protein, imaging data, pathology and radiology, evolutionary characteristics, host, and then we're going to end off on this really weird and fun story at the summary of all this. Okay, so this survival curve is the very first survival curve I saw in my life. It was the second day of my PhD, and I saw this, and a pathologist put it up to motivate a study we were going to do on chemotherapy for early lung cancer patients and whether or not it was going to be beneficial. And I saw it. And you know, it's a survival curve. So what you're seeing, let me get a pointer. So what you're seeing, of course, is the fraction of patients survive over time in the study. And as the study progresses, patients are dying of their disease. And I immediately said, why is there so much variability? Like, why are some patients dying after a few months and other patients are still alive 7, 8 years later? And the pathologist looked at me and paused for a second thinking, who the heck is this dumb student? And then said, we don't know. That we don't know has been the origin of my whole career, trying to answer that question, why is this variability existing? Because if you understand the variability, then you can start doing really important things. Because at time zero, you can group patients into those who need more treatment. Their existing treatment is not curative, or it's insufficiently likely to provide a cure. And equivalently on the back end, these are individuals who are overtreated. They've got either exactly the right therapy so that it's perfect, or they've got too much and we could save them some of the financial and medical morbidities of treatment. And so my goal has always been to figure out what's going on at and right before time zero so that we could make these decisions better. And so when I started my lab, all of my PhD work had been in lung cancer. So for lots of good reasons, you might want to switch, especially to avoid competing with your former supervisors. So I chose to switch, focusing on a cancer type. I looked at cancer types that had high incidence: prostate cancer, 1 in 7 or 1 in 8 men over the course of our lifetimes; actually relatively low morbidity, only about mortality, only 25% of prostate cancer patients will die of their disease. But because it's so common, it's the leading cause of cancer death in non-smoking men. So yeah, it's only 25%, but the numbers mean that there's lots of people dying from it and lots of people who are over and exactly correctly treated. Also, unlike almost any other cancer, we don't really have modifiable risk factors. The top are age, not modifiable, family history and African ancestry, not modifiable. And again, almost like any other cancer type, we are very, very, very good at early detection. This is a curve of the incidence in the population of prostate cancer. You can see it doubles around 1990. That's the introduction of PSA testing. $20 blood test, incredibly sensitive for prostate cancer. Not particularly specific, and that's why the mortality curve doesn't change all that much. But like, we're not missing large numbers of cases because we don't know they're there. We instead have a problem of identifying which ones are going to kill. And actually, that problem is so incredibly profound. In this disease, the standard of clinical care is for risk stratification is, as I mentioned, PSA testing, lots and lots of false positives. To assess where the tumor is and how large it is, imaging doesn't work well in prostate. We still don't have standard of care imaging in early prostate cancer. Instead, it's a digital rectal exam. A physician inserts a lubricated finger into a man's anus, palpates the prostate, trying to figure out if there's a bump. That's it. On top of that, biopsies. Standard, stick a needle into the prostate, take a small fraction of the tumor and see if of the tissue and see if there's a tumor there. The problem in prostate is that those biopsies are taken transrectally, which creates a 2% chance that a man is going to end up in the emergency room with sepsis, bacterial infection. This is not robust risk stratification from a patient experience perspective, but actually it's pretty remarkably good. Those characteristics allow you to stratify men into low, intermediate, and high-risk disease. And when I started in this field, I focused on intermediate risk localized prostate cancer. Very roughly, a third of these patients are being cured by existing therapy. A third are being overtreated and should actually be treated like low-risk patients, which means just watched. And a third are undertreated and actually needs adjuvant therapy, probably hormones, to increase their chances of cure. So you've got a perfect one-to-one-to-one risk stratification problem. Okay, at that point in time, you need to collect lots of data. So we started to put together a large national consortium, this is back in Canada, where we brought together the 3 major prostate centers and said, let's start answering really important questions. And the first one we asked was, is this problem possible? Like, can you solve it? And to do that, we asked, can you take small sections of the prostate and learn something representative about the whole tumor? Because of course, it does a man no good to say, your prostate cancer isn't going to kill you after you cut out the whole prostate. It just doesn't work. So you need to be able to take it off of biopsy sampling. So the prostate is about the size of a walnut. And what you're seeing here is basically like an egg slicer, layer by layer down the prostate. And so I'll zoom in on one layer. So in this layer, there's this little tiny region of what's called Gleason pattern 4, high-grade prostate cancer. There's a green region of Gleason pattern 3, low-grade prostate cancer. And so our study pathologist, Theo Vanderkwast, would go in and laser capture, microdissect, or in some big cases here, macro dissect large regions of tumor, and then we do all the molecular interrogation you could imagine. And the first thing that we asked was, is there spatial variability? In a single tumor, do things vary from location to location? Because if they do, then where you put a needle in really, really matters. And so complicated plot, let's just focus on this green rectangle. Each of these columns are 9 regions, one from each of 9 spatially separate regions from the same prostate. So 9 separate regions. And this top part is the number of protein coding point mutations, a low of about 20, a high of about 150. Right off the top, it's clear that different regions are different, right? You've got this order of magnitude variability in the number of mutations. That's true in every single patient that we look at. Here's another patient where there's one region with only about 20, 4 regions with 150 to 200. This is true if you look at protein coding mutations, non-protein coding mutations, epigenome translocations. It's just generally true. Prostate is the second most spatially variable tumor after virally associated liver cancer. But of course, your question is still going to be great, but is that clinically relevant? Like, does it matter? And so this plot will give a sense for that. This is the first man that I told you about, 9 regions from the prostate. You can see the regions anatomically laid out with some immunohistochemistry and H&E staining. And I'm just going to blow out this one tiny portion of the plot. So 9 regions laid out as columns. The first one is the diagnostic biopsy, which happened to be both the index lesion and the dominant lesion. So the lesion that was largest for this patient. This green rectangle for PIK3CA is a non-synonymous point mutation. It's H1054. This is actually druggable. So this is one of the 1%-ish of prostate cancer patients who have a druggable mutation when localized. So good candidate for, a basket trial. The blue dot for P53 is a single copy deletion of P53. Weak adverse prognostic factor, maybe a better candidate for surgery than radiotherapy, but really not telling you much. The problem, of course, is these other 8 regions over here, different parts of the same prostate, but that didn't come out on biopsy. And you immediately see only 1 of 8 regions had the PIK3CA mutation, so the chance of the targeted therapy working isn't much. The P53 is only in about half the tumor. We missed entirely on biopsy a 2-copy deletion of BRCA1, which is just bad news. If you superimpose on this the pathology, it turns out that the BRCA1 deleted regions are higher grade disease, therefore more likely to progress. And everything I just said is irrelevant because the patient had a relapse of the disease, and it was this region right here, number 3, which had none of the mutations that I told you about. So this is pretty frustrating. You've got a question where you want to be able to do an important risk stratification. You want to use molecular data, but it turns out that because of the spatial variability, you can't come up with a good biomarker, right? You'd have to basically remove the whole prostate to be able to do this. Fortunately, at exactly this time, we got very, very large funding to be able to do the thing that I had just shown was probably impossible. So that's great. And I didn't know what to do. So hired a really smart PhD student, didn't tell her that I didn't know what to do with this, and said, Emily, what do you think we should do? Emily Lalonde, who's now faculty at Wayne State in Detroit. And she said, well, obviously, I know what you need to do. You need to think about what happens earliest in cancer evolution, and you need to think about processes, not individual genes. If it's not common spatially, it's common because something underlying is driving it, and that's the process that matters. So it turns out that the type of mutations that happen earliest are copy number changes. And so this is a landscape of the human genome of prostate cancer, the genome of prostate cancer from chromosomes 1 to 22, blue for deletion, red for gain, and you can immediately see patterns, right? Different things that tend to happen more likely. Each row is a patient, and these patients look kind of more similar. Let me make that a little clearer. Here are the kind of 4 copy number subtypes of prostate cancer. Chromosome 7 gained, 8p loss, AQ gain, 8p loss, AQ gain. This is NKX 3.1, this is MYC. NKX 3.1 loss, a 16q loss, which encompasses a number of genes, and this very quiet subtype. The quiet subtype is the most common. Great. Does this matter? Well, yeah, it turns out to. One of the subtypes has much better outcome. The other 3 have poor outcome, well, with different kinetics of getting there, but poor outcome. And the subtype that doesn't have a lot of events, that's less lethal, it's the red one. Few mutations. So I just told you that tumors with few mutations have better outcome. I know you think that's obvious. I thought it was obvious. I made Emily look at 35 cancer types because I was sure this was a generalizable feature that everybody knew. Happens in 2, prostate and breast. It goes the opposite direction in ovarian cancer and is 0 or a weak trend in every other cancer type. So why this would be the case, who knows? But it means that you've got a great biomarker. You just count the number of mutations that are present in the tumor. We call it PGA, the proportion of the genome with a copy number aberration. It's just an acronym, whatever. But as soon as you divide the patients with lots of mutations from few mutations, you've already got a molecular biomarker that does a reasonable job of identifying patients with poor outcome. Okay, great. That's not perfect, but it certainly gives you some insight. Why are some patients having poor outcome, having lots of mutations, and other patients having few? What's the driver of that? We figured it had to be oxygen, DNA damage repair, ROS, and hypoxia. That's really important in prostate cancer relative to other cancer types, an established biomarker. And so we measured it. What we did was put needle electrodes into patients at the time of biopsy to measure physical oxygen tension. Those were then used to take the oxygen and the genomic measures and see how they're related. So what you're seeing here, PGA, genomic, how many mutations from high to low, HP20, basically the fraction of the tumor that's hypoxic from low to high. And you can see the profound zero correlation, right? That's a correlation of 0.04. There is no association, or, with large sample numbers now, it's a very weak association between hypoxia and genomic instability. They're just different. But actually, this is the last thing that Emily did that was really brilliant. She looked at this and said, yeah, great, but are they independently influencing patient survival? And when you put those two features together in the context of how long patients live, it's almost magical. Every time I look at this curve, it amazes me because here you see patients who have both high levels of hypoxia, very low oxygen in their tumor, and high genomic instability, and they have like a 50% survival rate. And here are the patients at the top with a 10% mortality rate or relapse rate that are the inverse, normal oxygen and few mutations, and in between the two one-off groups. And that's pretty amazing, right? All you need are these two events, and you don't get a perfect biomarker, but you certainly get a lot of insight because they're mostly independent. Problem is, you can't really easily put a needle electrode into the prostate at the time of every biopsy. It's a cumbersome procedure. So you need a way to measure it. And so a different PhD student in my lab, Vinny Bandari, took this up and said, does hypoxia leave kind of like a scar on the genome, a mark that tells you this cancer has seen hypoxia? And so to do that, he needed to have a way to assess hypoxia across a large number of cancer types. So he developed that method, and that's what you're seeing here. Hypoxia laid out from cancer types laid out from relatively low to relatively high hypoxia. You can see within each cancer type, huge variability from tumor to tumor. And you can see pretty encouragingly squamous cell tumors of the head and neck, lung, and cervix are the most hypoxic, which is widely known to be the case. Okay, great. So you can measure hypoxia, but what actually is happening? Are there specific mutations or is it broad trends? So let's take this top plot here. This is kidney clear cell, so CCRCC, tumors laid out from the most to the least hypoxic. So very little oxygen, normal tissue levels of oxygen. And here, this is deletion of CDKN2A, a very important tumor suppressor. And in red, lots of deletions in the hypoxic tumors and fewer in the normoxic. Okay, kidney clear cell is a little bit of a weird tumor type because of VHL signaling, which affects hypoxic signaling as well. Let's just take luminal positive breast cancer, a very classic tumor type. Again, most to least hypoxic, and look at P53 mutations, hugely enriched in tumors that are hypoxic. So it's not just generalized mutational processes, but individual mutations. And it's not just individual mutations, but it's core aspects of what happens. For example, here's a study done as part of a very large pan-cancer consortium from a couple of years ago. There's several thousand tumors laid out from most to least hypoxic. And you can see deletions are much more common in hypoxic tumors, but amplifications, duplications over here are much less so. So even certain types of mutations are different. Phrased differently, hypoxia completely remodels the types of mutations that happen in a tumor. And it's not just the types of mutations, it's when they happen. So we've got that same set of 3,000-ish tumors laid out from most to least hypoxic. And now I'm asking when the mutations happen. And you can see that they preferentially occur clonally, early in tumor evolution before subclonal diversification. And the number of subclones, the number of tumor populations, is the same. It isn't really associated to hypoxia. This is kind of a remarkable story. Imagine a tumor is a tree. A tree that is evolving in the presence of hypoxia looks like a palm tree. It's got a very long, skinny branch, and then at the top, it explodes. A tree that is evolving in the absence of hypoxia is shorter and will tend to have lots of branches going all at different places because it hasn't seen that selective pressure that chops off all the branches and forces it to get much taller. Okay, leave aside the really interesting things about hypoxia. You could now just measure whether a tumor has seen hypoxia by looking at the DNA alone. So you can get genomic instability and a pretty good estimate of hypoxia from just DNA copy number measurements. Emily put that together into a biomarker. It's got about 80% accuracy. Any great reviewer says, cool, validate that. So we worked with a company called GenomeDx, then in Vancouver, now in San Diego, that does this type of work in a 1,000-patient cohort. And you can see the AUC, a measure of accuracy, is about 80%. That's great. All of these are other clinical or commercial or academic biomarkers that are used. And you can see we're a little better than everybody. So that's good. You might then ask questions like, does this work in other risk groups, low-risk patients or high-risk patients? It does. But as a good academic, you also ask the question, well, 80% is really nice. What's going wrong with the other 20%? One arm of my lab really thinks about the implementation work behind this 80%. That's really good. I'm going to talk to you about the 20%, what's missing, and I'll tell you a few stories of negative data because we have a lot of stories of negative data that don't explain the 20%. The first thing you might think, and Paul certainly thought, was, yeah, okay, it's got to be the fact that we didn't look at the rest of the genome. We just looked at copy number aberrations, and actually we just looked at the nuclear genome. So what happens when you think about mitochondria, point mutations, translocations, that sort of thing? So we did that. We took hundreds of localized prostate cancers, did whole genome sequencing on them, and we discovered fascinating things. Just give you one quick example. These are localized prostate cancers, and we're looking at the number of point mutations in the 3 billion bases of the genome. And you can see most of these are in the low thousands. By contrast, a breast cancer would be about 20,000, a lung cancer about 200,000, a melanoma in the millions. So it's low. But what's amazing are these outliers. Here are tumors that have like 10 mutations in the 3 billion bases of the genome. How is that a cancer? How can only 10 things be enough to transform something? And you might immediately go, sure, that's point mutations, but I bet they have a lot of inversions or translocations. Nope. Maybe they have lots of copy number aberrations. Nope. These are just quiet genomes that don't have a lot of things going on. And this is something that we're really interested in. How do these things become cancer? And there's a lot of things that we're doing to try to understand that. You can also, of course, use these data to discover features that drive localized prostate cancer. And so we've got this set of 40 features that we've identified that are drivers from the first 200 tumors that we sequenced. They include well-known cancer genes like ATM, P53 is on the list here, down there. And each of these is associated with either the incidence or the lethality of prostate cancer. But you might then go, what happens? Did you have enough? What happens if you scale to doing hundreds more? So this is recent unpublished work from a large number of people in my group and friends in Australia and back in Toronto, where we looked at a cohort of 650 localized prostate cancers. And from that, a grad student, Helen Zhu, back in Toronto, was able to identify 200 separate driver events. We found 40 with 200, 200 with 600. We're nowhere close to saturating. There's this really long tail of important but rare events that are driving cancer initiation and progression. You might ask, okay, can you put that all together into a biomarker? Well, we certainly tried, and as best as we could get, we got a biomarker with an AUC of 0.83. So I'll highlight what we did here. We took Emily's really nice $150 copy number assay and turned it into a $10,000 sequencing assay that is not more accurate. So that didn't work too well. In fact, it's even worse than I described here, because even easier things to predict than lethality aren't really easy to predict. You might, for example, take grade, the way the cells look under the microscope, measured by pathologists every day, thousands of times for prostate cancer in any given city. And here we're taking low to high grade prostate cancers and different types of mutations. And certainly in the way it looks for this paper, there's a increase in the number of mutations as you get to higher grade. But if you take a look at the actual magnitude, these are Z-scores. It's like 2 standard deviation difference, 3 maybe. That's not a biomarker, right? Like you can't convert that into something very accurate. And you might ask, are there more driver mutations? And there sure are. Low-grade cancers have about 5, high-grade cancers have about 10. But look at that, there's so much overlap. There are high-grade cancers with only 1. So you can't convert these things into strong biomarkers, even for an easy endpoint. And you might say, okay, but maybe there's one specific mutation which is really accurate, and there's not. Take a look at a mutation like FBX031, where it occurs in 30% of low grade and what is that, 50% of high grade? It's a signal, but it's not enough signal to be able to make a great biomarker. That's why we're getting these AUCs of 0.8. Okay, great. So more negative data. Let's think about the rest of the central dogma. What else is going on? You might look at the epigenome. We looked at the methylome, H3K27 acetyl marks with the first ever primary ChIP-seq that was done. And when you do it, you find subtypes of disease, associations between somatic mutations and methylation subtypes, and you can put it together into a biomarker. So I'm not giving you the AUC here. We didn't put it in the papers because it is well below 0.8. Actually, it doesn't make things better at all. You then might say, okay, it's not the genome or the epigenome, it must be the expressed genome, the transcriptome and the proteome. And I'm doing 2 beautiful collaborative studies here, no favors by describing them very quickly, but we took 160 tumors and did ultra deep RNA sequencing, 1 billion reads per tumor. And we did whole cell proteomics, identifying 7,000 proteins per sample. And again, we found fascinating things, like things that just make you go, there's so much biology to discover. My favorite is the fact that protein and RNA are not fully unrelated, but basically unrelated. This is the 10% most abundant proteins in prostate cancer. And each dot is a protein, and the y-axis is how well the protein and RNA are correlated. And the median is 0.3. That's a correlation, which means that the amount of variability explained is basically the square of that. 10% of the variability in protein levels is explained by RNA levels. It almost makes you wonder why we do RNA sequencing. Like, it's just so non-explanatory. And actually, that's a good thing from the point of view of biomarkers, because if we take individual genes and ask, are they a biomarker at the protein and RNA level, they're not well correlated. There's lots of examples of genes like MDK here, which is an RNA biomarker, but not a protein biomarker. This is nice. At most, it explains 4% of the 20% that's missing. We're not getting anywhere. At this point in time, I was pretty unhappy because nothing was working. So I did what I would always do in those cases, which is go for beer with people who know more than me. And so I went for beer with people who diagnose prostate cancer for a living, pathologists and radiologists, asking, what do you see? What's weird? What might be going on that I don't think about? And one of the study pathologists brings out his phone and goes, hey, look at this. Don't these 2 things look differently? And even I could tell that that and that look really different. This is a single prostate cancer. This is acinar adenocarcinoma of the prostate, and this is intraductal or cribriform architecture of the prostate, IDC. And they look different. And they said, maybe they are really different in other features, the molecular or the clinical or things like that. And that led to a long series of studies. And I'm going to subset a couple of these to show that it's associated with poor outcome. Tumors that have any of this intraductal carcinoma phenotype, even in a biopsy, have very poor outcome. They turn out to be more genomically unstable. So these tumors have more mutations than the tumors without that weird pathology. And at this point in time, I was just moving to UCLA. And I told this story to Rob Reiter here, who's one of the urologists who leads the prostate spore. And he said, hey, I wonder if this is related to whether or not these tumors are visible under MRI. And this was the first study that we had come out of the UCLA work that we did. And tumors that are intraductal carcinoma positive are 7 times more likely to be visible under MRI. So you've got this huge increase in whether or not they're radiologically evident. And ultimately, that led us to a series of studies to understand their molecular features. And in a really nice collaborative UCLA Toronto study with Rob and a proteomicist in Toronto, we were able to come up with biomarkers that predict visibility from the proteome and can use that to be a semi-accurate prognostic marker. And this is what clued me off on what we were doing wrong, the whole core of it. Because everything I showed you here was, and we took a biopsy, and we took a single sample. That's dumb. Cancers are an evolving mutational feature, and you don't just need to know where they are, you need to know how fast they're moving. We needed to be able to infer aspects of tumor evolution. How do you do that? I had no idea. So I got together with a bunch of friends who are smart at that kind of stuff and said, how do we do this? This is David Wedge, who was then at the Crick and is now at Manchester. Quaid Morris, who is then in Toronto, is now at MSK. And Peter Van Loo, who is previously in the Sanger and is now at MD Anderson. We all left before we could finish this project. The idea of it was, can we estimate tumor evolution? We put together a crowdsourced benchmarking challenge. We said, we don't know the answer, but we have some ideas. Let's make it available to groups around the world and ask them to give us their best ideas so we can use it to solve our important problem. And we defined tumor evolution according to 3 basic questions. What is the tumor made up of? What cell types? How many of each? What mutations go together in a single cell? What are the populations of cells and their mutational composition? And how do those relate together in a phylogenetic tree? We then said, okay, let's put together this crowdsourcing challenge. We started it in 2014. The last paper is in revision now, so like it's almost finished. I could tell you so many stories of all the things that go wrong in doing this, but it ultimately allows you to define a standard for a field. And it's remarkable the number of things you discover nobody can agree on. My favorite example is nobody could agree on how to compare 2 phylogenetic trees and say which one was worse. So imagine that you had a true tree and 2 mistakes. How do you decide which of the 2 mistakes is worse? Nobody could agree. We ended up getting a panel of 9 experts in the field, asking each of them to look at hundreds of trees and go, here's the truth, here is 2 different mistakes, which mistake is worse? And then back inferring from their human intuition, the mathematical structure that they were talking about. So give you an example, here's the true tree. Here is one possible error where the bottom node is split into 2, so they've got an extra subclone. Here's a different error where the bottom node and its parent are now siblings, so parent is grandparent. And then each of us went through and voted whether we thought this was a bad or less bad mistake and ranked it. And then we used that to infer metrics that resembled what we thought. We did not always agree. And actually, like, the disagreements are so fascinating because they really relate to what cancer types we study, what types of problems turned out to be more problematic for that. In the end, after massive work, different PhD student, Adriana Salcedo, was able to turn this into a demonstration of what best practices are in the field. We had on the order of 30 teams participate from around the world, and we were able to identify, we don't call them winners, but best performing teams that allow you to figure out the standard of how you should do this. And that actually gives remarkable methodologic insight, right? By knowing this, we were able to figure out things that people did wrong. A great example is work that a different PhD student in my lab now, Helena Winata, is looking at, which is one of the core assumptions of all of these methods is that everybody starts with a single subclone that gets more and more complicated. It's a terrible assumption. Nobody's ever found a tumor which has only one population. They just don't do that. They evolve. And you can see how complicated tumor evolutionary trees can look. They just have remarkable number of nodes and complexity. And the deeper you look, the more you find it, which is why you need to improve methods. Okay, back to prostate cancer. How do these things evolve? Well, great. We looked at this in a cohort of just about 300 localized prostate cancers. This is the normal. The branch lengths are the number of mutations. And you can see about 120 35% of tumors are monoclonal. In the index biopsy, in the place that was first detected, there's only a single population. Of course, if you looked at the rest of the prostate, there's lots more. This is just giving you a sense for how complicated it is. In the other 200-ish, they're multiclonal, polyclonal, some with 2 populations, some with 3, some are branched. Interestingly, all of these branched ones are BRCA2 germline positive. All right. Does this matter? Well, it really matters. When mutations happen, tells you a whole lot about their behavior. So here we're taking a look at localized prostate cancers, and here at metastatic prostate cancers, big numbers, really big cohort. And you can see, of course, most mutations are more likely in metastatic prostate cancers, but actually some are not. These are tumors that they cause a cancer, but the cancer is not very likely to metastasize. And when a mutation happens, really influences survival. This is a major oncogene, and you can see tumors without a mutation in it do well, I mean, not really well, but reasonable. Tumors with a mutation that's only in a subset of cells of the tumor, it's branch or subclonal, do intermediate, and tumors with a mutation in every cell of the tumor do worse. That's MYC, that's P10, you know, basically the most established tumor suppressor and one of the 2 or 3 most established oncogenes, other way around. So that's really encouraging. And actually, if you put this together into a biomarker, you can get really remarkable results. What you're seeing here is intermediate risk localized prostate cancers. If this looks like the very first survival curve that I showed you of prostate cancer with low, intermediate, and high risk disease, it should. It basically looks like that. But at time 0, they were all intermediate risk. And that top group are tumors that are monoclonal, normoxic, genomically stable, MRI invisible, intraductal carcinoma negative. And the bottom with 70% relapse rate is the inverse. This is basically the answer. If you call this in percentages for a biomarker, it's 95, 97% accurate, right? Like this really, really works. And we're now doing a very large 1,000-patient validation study using the UCLA Spore Biobank, sequencing everybody to demonstrate that this really works because it explains the missing variability. 5% missing, that's probably clinical problems with annotation of the tumor, positive margins, other undetected things that we wouldn't be able to work through. So we think this is about as good as we'll get. Okay, so I'm going to then spend a couple of minutes telling you what happens next, which is, yeah, you implement, that's one arm of it, but you still get excited and interesting research questions. And I got, yeah, this is why they're all different, but like, why? Why did this person have a tumor that went this way and somebody else have a tumor that went that way? These are the processes, they come together to cause lethality, but why? And so that got me thinking about host. And I'll show a few examples of just how important the host genome is in cancer, and then motivate some of the things that we're working on now from that. The first thing I asked is, what's the biggest host difference you can get? Sex, right? Like it's a whole chromosome, it's big. And I'm showing some examples of a couple of different tumor types: AML, kidney clear cell, head and neck, stomach and esophagus, and sarcoma. We have lots more examples. These are just a few. Take a look at sarcoma. If a sarcoma arises in a woman, it has about a 7 or 8% larger fraction of the genome mutated. It's a whole chromosome. An entire chromosome difference in the number of mutations that occur in a sarcoma arising in a woman than in a man. By contrast, AMLs, a liquid tumor arising in women, have very few. The median is 0 mutations, copy number mutations. And of course, in men, the median is a little larger and it's about 4 or 5-fold higher overall. Big, big differences. Okay, so sex is large. Can we find something that's a little smaller? Sure, let's take a look at ancestry. A large number of SNPs that are co-occurring. If sex is a whole chromosome, ancestry is on the order of hundreds of thousands, kind of point mutations, point variants. And so here we're taking a look at mutational processes. So this is the BRCA phenotype, and you can see that it increases in different groups, highest in individuals of African ancestry across all cancer types. This is an insertion deletion process associated with human aging, and you can see it is lowest in individuals of Asian ancestry. I'll give another example, my favorite one. SBS41 is a single base mutational signature. Nobody really understands what it is. It occurs in 60% of breast cancers arising in women of Asian ancestry and almost never in any other woman. Why? Nobody knows, but there are these large differences from cancer type to cancer type in how ancestry influences cancer evolution. Okay, well, maybe we could look at something besides ancestry, like just your age. So if you take a look across cancers, and this is about 15,000 tumors, so it's a large cohort, you can see that there's a slight positive trend that as somebody gets older at the time of diagnosis, there are more mutations in their tumor and more of them actually occur early in tumor evolution. But now if you look at different cancer types, in medulloblastoma or stomach cancer, those trends are much more pronounced. And in melanoma, it goes the opposite direction. Age influences the number of mutations, mutational processes, drivers, everything else that you could like. Okay, what about individual germline SNPs? Like, can you just look at a single point in the genome and figure something out? Really brilliant grad student with me, Katie Houlihan, looked at this stuff for her PhD and 3 really landmark papers that are making their way out now. And the answer is depressingly yes. Here is a single SNP, and we're looking at one mutation. It's a point mutation in FOXA1. If the individual has 2 wild-type copies of it, they have a 10-to-1, so 1 in 10 or 1 in 11 chance of having this mutation. If they have 1 alternative allele, it's 1 in 20. And if they have 2 alternative alleles, it's 1 in 100. You might go, okay, Paul, statistical noise. Here's a validation cohort, 1 in 10, 1 in 20, 1 in 100. A single region in the genome, one single base, is changing the probability of this mutation 2.5-fold. Or phrased differently, if one was to take a look at this individual at birth, you can make a 60, 55% likely estimate that if you have their genotype, will or won't they get the specific mutation if they get a prostate cancer, which is remarkable, but also kind of depressing because now everything I've told you about, you can't change. So what is my lab focused on now? I'll give 2 quick vignettes. The first is, what are the things that we can change? Exercise, exercise, exercise. We're doing this large series of clinical trials of exercise where we're taking patients in the window setting or the survivorship setting. So window between diagnosis and treatment and putting them on intense exercise protocols. They get a treadmill shipped to them. They, on the treadmill, do Zoom-based video exercise sessions with an exercise oncologist. And then we get weekly blood draws by sending a phlebotomist, mobile phlebotomist to their home. We get weekly stool samples, pre and post genomics on the tumor from the biopsy and the surgery. We ship them a watch so we can get their mobile health data. And then we can look at what the differences are. The first paper from the clinical trial is out for review right now. All I can tell you is it will make you exercise more. Like it really, really does. The other thing that we're doing is realizing that there are so many confounders that you need massive sample sizes. And this is actually, of course, one of the key reasons I came to UCLA. This is the ATLAS cohort run by the Institute for Precision Health, which Clara is the deputy director for. Its core idea is to have a very easy consent process, a 5-minute video consent that consents patients for use of remnant samples, additional blood draws, de-identified use of medical records, and now for more recent patients, return of any genetic results for clinical actionability. Available in 2 languages with 4 translations, and there's a pediatric consent video that's under development right now, actually as part of the CTSI renewal. And the first set of 45,000 exomes generated from that is going through analysis right now, just very preliminary, looking at pipelines and ancestry and stuff, but gives you a sense for the scale at which we're going to be able to need to look at things to address all those host level confounders. Hundreds of patients won't cut it. Thousands of patients won't cut it. It's really millions. All right, so I promised you a really weird story at the end. So let me summarize with something really, really weird. Everybody has heard that you must publish or perish. If you don't publish, you can't survive in academia. And so a good friend of mine, Chris Hovens, who is co-lead on some of the prostate studies, we were sitting at Ministry of Coffee here in LA when he was doing a sabbatical with me. And he said, Paul, I just saw this paper that was retracted. And I said, cool. He goes, but I thought this person passed away. And I was like, they did. And he said, how did they sign the retraction notice? I said, that's really weird. We should look into that. And like any good scientist, we went ahead and asked how many papers have deceased authors on them. And there are shocking trends in the number of publications that have deceased authors. Before 1998 in PubMed Central, which of course is not all of PubMed, it's a subset with open text and stuff, we found 0. And then the number ever since has been increasing every single year, initially exponentially, now just linearly. It's increasing faster than the rate of new publications or the rate of co-authorship. And if you make a prediction from any timeline, you'll be wrong. Like it just keeps on going up faster and faster than you think. And you might ask, like, when does this happen? Well, here we're taking a look at the number of publications that a deceased author has done. And we validated these using death records and other sources of data, and the fraction of their papers that occurred after they passed away. You can see outliers like this. This is somebody who published 170 papers after they passed away. You can also note over here, individuals where 100% of their papers occurred after they passed, some with as many as 20 or 30 publications. And now you're going to ask, what's the timing of this? Year 0 is the year of their passing. And there is a peak in publications in the year that they pass away. Okay, the way we actually kind of think about this, leave aside joking, is, this is saying something fundamental about authorship. What's to stop me from including Albert Einstein on all of my papers, right? Nobody checks this stuff. He's a deceased author. Criterion for authorship includes stuff like agreeing to the final paper. But of course, we have publications that take 8 years to happen, as I showed you. How do you acknowledge your colleagues who are critical to it and yet make sure you're staying in touch with guidelines for publication? This is actually a big and, as you can see, increasing issue. And the community has come to the idea that we really, really should acknowledge those authorships. And yet there's no actual journal guidelines. Pretty much every single one of these papers is in violation of ACMG guidelines. Also, the peak at the year of passing seems to indicate something else. All right, now let me summarize this. There's a ton of things that influence the metastatic potential of a cancer. Genomic instability, the microenvironment, histology, evolution, specific genes, host behavior. And these are prostate cancers. There's a lot of other things that would influence other cancer types. You'll notice that I didn't talk at all about the immune system. It's just not important in localized prostate cancer. But in other cancers, it's a central driver of early lethality. So it's not that we think it's the same processes in each cancer type. It's a subset of maybe 20 or 30 processes. A subset are important for each different cancer type. These things are correlated partially. And the data kind of suggests that you need composite biomarkers. You need to merge a lot of things together. And actually, the other big direction of my team is doing all of this in urine in a non-invasive way. And we just have a large-scale collaborative funding to do 2,000 urine proteomes to try to really understand, can you do this maybe even at home with a simple at-home urine test? Okay, so I did none of this. This was a huge team consortium. This is a subset of the people that we've been working with here. I particularly note Tom Kisslinger back in Toronto, who took over all of the prostate leadership work after I left there, but also critical collaborators here. And that's my team who actually does all the work. I'd be happy to take questions. You can come up if you have a question. And I believe, yeah, come on up. Okay, one sec for the people online. We'll have a mic in a sec.
Yeah. Right, here you go. A part of the presentation, you showed that age was a good predictor for a lot of the mutational burden disease and tumors. How might that differ in biological age? Because I know that Steve Horvath used to be here and Morgan Levine, I think. They came up with this great way to quantify biological age differences outside of chronological.
Yeah, it's great. Yeah, it's a great question. I'm not sure if everybody online heard it, but it was, is age a great predictor of mutational burden? And would you be better off using biological age inferred from, for example, the epigenome? The answer is two parts. One, age isn't a good predictor. It's correlated and it makes your life difficult. But actually, if you turned it into a biomarker, it's like 55% accurate. Like it's got signal, but it's not good. But it's enough signal that it can really annoy and confound more complicated things that you want to do. Anyway, that wasn't your question, which was really, is biological age a better predictor or not? Nobody's looked at this systematically, and it's a good option to do so. But in prostate cancer, biological and chronological age are very well correlated, much more so than in other cancer types. Unclear why that would be. The best guess is that it has something to do with the mutational processes that initiate prostate cancer, and that those being more associated with just the number of cell cycles than with a specific exogenous stressor or things like that. And that would be supportive of the fact that there aren't many modifiable risk factors, right? Smoking doesn't really strongly affect prostate cancer. Obesity doesn't strongly affect prostate cancer. Even diet isn't really a strong effect. Exercise is the best that we've got, but it's not very strong. So in that context, it could just be the cancer type. We don't see it there. Nobody's looked pan-cancer that I'm aware of, and it's actually a pretty good and easy project given TCGA data. Other questions? Yeah.
Hi. You have mentioned about the mutations and age factors relating to the progression of the cancer. Have you done any studies relating to the intracellular organelles in relation to these?
Great question. So we looked at intracellular organelles. Yeah, a couple of things that we've done exactly on that. So we did a number of studies with fractionated proteomics, where we've done proteomics of the bulk, but then extracellular membrane, nuclear fraction, different organelles, 3 or 4. And the net effect of it was that there were clear differences, but they weren't the kind of differences that worried you about anything. The prognostic genes remain prognostic. There was a few extra hits, but not much that made you really think about it. We then did a large scale mitochondrial study, which I didn't show here, actually a pair of them. And they showed really important things in the mitochondrial genome, but not a lot of particularly interesting things in the transcriptome or proteome on top of that. That's led us to deprioritize organellar assessments overall, just given all the different things that we're doing. With a couple of exceptions, in model systems, we do a whole bunch of it. And the other one is that we're doing a lot of work in urine on extracellular vesicles, taking a look at those of different sizes. And it turns out that extracellular vesicles that are big versus small represent different organelles internally. That's not why we were looking at them, but it turns out that we've been non-invasively sampling different organelles in different ways without really intending to, and that's led to some interesting biology. So not a big focus for these questions, but interesting for other reasons. Thanks. Yeah.
Thank you very much for the great talk. So I'm really very interested in your, you mentioned that the hypoxia, it is the good marker for the tumor evolution or might be more aggressive. So as you know that the cancer, they have Weber effects, and then they use glycolysis in the presence of oxygen supply. And they might also use the vasculature or angiogenesis in the inner side of the tumor. So I'm really interested, do you have any analysis or prediction of what is changed about Weber effects in the tumor? And also, does that correlated with the size?
Good question on the size. So yeah, it's a great question overall. Tell you a story. We have an angiogenesis paper, and it turns out to be a breast cancer angiogenesis paper because the student who was working on it did a whole bunch of work, and there was like zero association of angiogenesis with anything in prostate cancer. And then it was like, oh, we should see if that's general across cancer types. He applies it to a breast cancer data set, gets a really interesting angiogenic signature, interesting correlatives. And that was like a PLOS biology paper or something a few years ago. It's just not there in prostate. No particular guesses as why, but that's the case. Then second part was Warburg effect. Yeah, a lot of people hear about the mitochondrial work and the hypoxia and ask, how are those related? There's nothing. There's really nothing. And even in prostate, we can't pick out a clear Warburg signature associated with hypoxia. I'm not sure what that represents. My best guess is that what hypoxia is really doing in prostate is more related to its correlatives, acidosis, or interstitial fluid pressure. So I'm willing to bet that hypoxia is creating a more acidic is creating a more acidic microenvironment, and that's impacting DNA damage repair and other features rather than hypoxia itself through Warburg or other mechanisms. Pure speculation, nobody knows, but yeah, neither of those has been promising. Last thing I said when you talked about tumor size, we've done a bunch of studies of that now, not super well measured, but as best as we can tell, the genetics associated with tumor size are different from the genetics associated with tumor grade or the amount of PSA that's being emitted. So it does seem to have a different genetic signature, but it's very weakly associated with hypoxia. It's like one of those, it's got a correlation, but it doesn't matter kind of things, and is not associated with angiogenesis, proliferation rate, anything else. So yeah, it's interesting what might be going on there. Good question.
There's a question online. I believe the question is prostatic. What is the relationship between prostatic hypertrophy and cancer?
Yeah, so good question. There's a number of benign prostatic diseases or afflictions, the most common of which is benign hyperplasia, BPH of the prostate. A group in New York, Chris Barbieri, looked at that in great detail and like sequenced hundreds of them, did DNA and RNA and all of that stuff. And they're like basically unrelated to prostate cancer. Like these things seem to have no association with prostate cancer. There are also some pre-malignant features like PIN, prostatic interplastic neoplasms, and they are kind of weakly associated with prostate cancer, but not in a deeply meaningful way. And my favorite example of that is if you take like a region of normal prostate and sequence it, you find just as many mutations as you do in a PIN, and sometimes even as many as in a prostate cancer. So like, it's more like local prostate just tends to accumulate mutations as a man ages, rather than PIN or BPH are precursors or cofactors or confounders for prostate cancer. And last thing I'll say, then I think we're out of time, is Raziel's group from the UK, from ICR in London, did a beautiful study, 50 patients where they did multi-region sequencing of the normal prostate, looking at the DNA, and there are hundreds or thousands of mutations that have no relationship to the tumor. It's like the normal prostate is just accruing mutations all the time, but if it doesn't get the right mutation early, our work, then it won't become a prostate cancer.
Well, thank you, Paul. We're out of time, but people online would agree that this was an outstanding talk, and we want to really thank you for your time and for sharing this with us.
Thank you so much, everybody, on and offline.